Okay, so the title of my talk is which size matters when? Is alternating macroevolutionary regimes of Phanerozoic marine animal body size, and this title focuses on the work that I do at Stanford. But what I do at Stanford is not actually what I do normally. And so what I thought I'd do first is go over a little bit about my background and what I do as a research scientist, and show you some examples of how I think about the fossil record and how to approach it. And then ultimately, I'm going to come back to this body size project I'm working on, and to show you how it's all ultimately linked. And so, so as a research scientist, I consider myself a stratigraphic paleobiologist. And so throughout my entire career, I've been heavily involved with a lot of paleontological work, but I also do a lot of stratigraphic and sedimentological work. And so one way that I also would describe myself is that I am a stratigrapher that's trapped inside of a body of a paleobiologist. And so I do both, but I've been doing more of the paleobiology, mostly because of the programs that I've been involved with do more of that, but I also have an equal interest in sedimentology and stratigraphy. And so throughout my career, I've been very fortunate to be able to do field work and research basically all over the world. Here's just a few photos of fossils I found in the field and some of the cool experiences I've had. One of the cooler ones that you can see here, a photo of myself at the Panama Canal, where I spent a year, where my job was to collect the vertebrate fossils, but research-wise, I spent most of the time measuring stratigraphic sections to create and develop the depositional environmental systems that characterize the marine units throughout this part of Panama. And so some of the tools that I use are primarily, as I mentioned, I try to create the context needed in order to understand biological patterns and evolutionary dynamics in the fossil record. And so I do that with a combination of stratigraphy, of sedimentology. I use petrography. I'm more trained on the carbonate side of things, but I can also do a little bit of siliciclastic work. And once I create the context, then we can go in and understand what the biology is doing, how are the evolutionary dynamics working out, and how are they being driven by the stratigraphic record. And so one way that I can also characterize my talk, and just as a sort of a alternate title or like a subtitle or underlying theme, is stratigraphic controls on biological signals in the fossil record. And so what I'm going to go through today with y'all is show you a couple of examples and how understanding or having an understanding of the sedimentological and stratigraphic controls in an area may affect how we interpret biological signals, and why it's important to be able to have that context in order to assess these patterns. And so first off, I'll talk about a little bit about my work that I did during my master's on assessing a mass extinction event. And then I'll talk a little bit about my dissertation work at the University of Georgia, where I looked at ecological composition during the Jurassic. And then finally, I'm going to round back to what my original title is, and looking at body size patterns throughout the Phanerozoic, and how having an understanding of stratigraphic controls ultimately affect how we view and interpret these body size patterns throughout the Phanerozoic. So to start, I'm going to start on my master's work here. And for my master's, what I did was I focused on the late Mississippian, early Pennsylvanian interval, in which, so time interval in which recently, over the past 20 years, workers have argued that there's a fairly large mass extinction event. It may not be necessarily as big as the traditional Big Five that we all have come to know, but it's still considered a pretty substantial mass extinction event. And so one of the things about this mass extinction that's sort of controversial is that if you look at it from a global perspective, you definitely see that there's evidence of an extinction event. You see there's a drop in diversity across many major marine groups during this time interval. But when workers have started looking at this from in North America, they started noticing a pattern that doesn't quite match up with the global signal. And so in some places, particularly in the Appalachian Basin, in the Mississippian Basin, in Illinois, and also in Arkansas, some work there has demonstrated that diversity hasn't actually changed, doesn't drop much across this interval in the mid-Carboniferous about 330 million years ago. And so I wanted to expand on some of this work and to test whether or not I see evidence of a extinction event across the Mississippi and Pennsylvania boundary at the global type section for this interval. So a global type section is basically is considered the best place in the world that represents a certain geologic time interval. And so in this case, the global type section for the late Mississippian for the Mississippi and Pennsylvania boundary is in Nevada, it's in southern Nevada. And I have a map here. And so it's about 40 miles northeast of Las Vegas. It's fairly easy to get to. Arrow Canyon is a very famous and well-known locality because it's a global type section, but also to show you from my first figure here, the cyclicity here of deposition is amazing. And so what you can see here, it's just amazing cycles of carbonate deposition over and over again. And what's driving this deposition, and also what people think drove the mass extinction event, was the onset of the late Paleozoic ice age. And so the late Paleozoic ice age is considered to be the longest ice age event of the Phanerozoic that started in the late Mississippian and went on until the early Permian. And so here at Arrow Canyon, you see the rocks here record that onset of the ice age, and it's really cool. And it's one of the best places to go see that. And so sea level here changed so drastically that in many places, and unfortunately I don't have photos here to show you, in many places you see stumps and roots of trees preserved on limestones. And so these limestones that are marine, full of marine fossils, were exposed long enough that trees were growing on them. But they didn't last very long because sea level rose again and basically flooded everything. And this happened over and over again in this interval. So it's a really cool place to assess whether or not you see evidence of a mass extinction event. And so, so this is basically where I focus for my attention for my master's work. And so I mentioned the mass extinction event. The group that I targeted were primarily brachiopods. And so brachiopods were a primary group that are thought to have been hit hard because of the extinction event. And so I focused on them. And so in order to test whether or not I see evidence of a mass extinction event in brachiopods, I sampled for brachiopods below and above the Mississippi and Pennsylvania boundary. So on the left, you see the Mississippian section from the canyon. You'll see that it's a little bit, there's a lot of covered intervals. These intervals are primarily shales, and we think that these represent these intervals where sea level changed very quickly, and you had limestones that are covered by terrestrial silt and muds, and sometimes you get paleosols. But as you go up the section, you lose these more shaly intervals and it becomes almost entirely limestone. And so my approach to testing for the extinction event was to sample 5 beds below the boundary and above the boundary. And within these beds, I sampled multiple replicate samples. And so I took, I think, about 3 or 4 samples per bed. And so in total, I had about 35 samples to compare between the Mississippian and the Pennsylvanian. And so one of the other things too that I wanted to do is I wanted to sample within similar lithologies so that we can get a control for facies. And so because the environment here is changing very quickly, water depth is changing very quickly, it's crucial to be able to compare among facies to get a true picture of how diversity is doing. And so just to show you here quickly, these IS samples refer to the formation. And so in the lower Mississippian, I sampled the Indian Springs Formation. And the upper part of the Mississippian is the Birch Spring Formation, which then goes all the way into the Pennsylvanian. So if you see these IS samples, it refers to the Indian Springs, and BS refers to the Birch Spring. And then the color-wise, so if you see the red, any red sample was collected in the Mississippian, and everything in blue was collected in the Pennsylvania. And so now on to results. And so first thing I did was I took the raw diversity to compare how it changed throughout the section. And so here I have a couple of ways that I measure diversity. First, I have raw richness, which you see in the black curve. And raw richness is just basically the raw count of different taxa in these samples. In this case, I focus on the genus level. So these are the number of genera of brachiopods in these samples. And so at a quick glance, just looking at the raw richness, you see this huge drop in diversity. And so at first glance, you might think, oh, maybe here's where we see the evidence of the extinction event. And so once we took a look at the raw richness, I also applied a couple of quantitative methods that controls for sample size. And so even though I took the same number of samples, the number of individuals within those samples varied. And so these methods account for that. And so the first method I used was rarefaction. And it shows an overall similar pattern as you see from the raw richness. So you see that there's a huge drop here at the end of the Mississippian before it rises again in the Pennsylvanian. But then I used a more involved method called shareholders quorum. And when you look at that, you see that diversity actually stays pretty flat, which is pretty interesting. And so because we have sort of a differing view of how diversity changes across this interval, the other thing that I did was I employed multivariate techniques to assess the compositional differences among samples between the Mississippian and Pennsylvanian. And so by that I mean that each sample has a certain set of genera that are found there. And so we wanted to see was how different are they from each other, from the Mississippian and from the Pennsylvanian. And so for the first part of assessing whether we see changes in compositional taxonomic composition, was I used this method called clustering analysis. And so a clustering analysis, what it does is it groups the samples that are most closely similar to each other and creates basically a dendrogram or a tree, very similar to what you see with a tree of life, for instance. And so here on the left axis, on the y-axis, excuse me, you see the sample names. So again, IS for Indian Springs Formation, BS for Birch Spring, and the colors represent the time interval, so red for Mississippian and blue for Pennsylvanian. So if we take a quick glance, you'll see that there's two main clusters that form. There's one here, and then one big one down here. So I'll highlight them there. And so again, at first glance, you see that there's definitely a difference in composition. And so if I go back to the diversity plot, so again, so just notice the IS1, IS2, and IS3, this sort of corresponds to this drop here. And so I was like, okay, well, maybe the fact that diversity drops and you see a change in composition, maybe there is evidence of an extinction event there. And so the next thing that I did was, okay, well, this tells us very broadly how similar the composition is among these samples, but also use a similar method called non-metric multidimensional scaling that does the same thing. But what it does is it gives us a chance to try to figure out what's driving those differences among these samples. And so this is what that looks like. And so again, you have the Indian Springs, Mississippian samples here, and then most of the Birch Spring with samples over here. So at first glance, again, you see a similar pattern. You see a big difference between the IS samples here and the BS samples over here. And so to make it easier, you can highlight them here and the circles. And so what you can see here is that there's quite a bit of variation among this group. And so even though there's a big difference between these two formations, I wanted to also assess what's driving the variation in here. And so looking back on the samples and looking back on in the field, we find out that what's driving the variation in these samples can be both water depth and the amount of silt in the samples. So basically, the difference you see between these groups of samples is driven by the amount of silt and deeper water. And so even though I tried to constrain the water depth by collecting among the same species, I failed because in the uppermost samples, well, I tried to collect similar lithologies. When I went up there, it wasn't fossiliferous. And so one of the issues was in extracting these fossils, these were all silicified brachiopods that involved dissolving blocks of limestone to get out the individuals. And so when I'm in the field, oftentimes I cannot tell what's in the rock. And so I will often be disappointed when I come back and notice that the samples didn't have any fossils in them. I have to go back and resample. So I had to sample a different lithology just slightly above where I initially sampled, and it turns out it was just slightly deeper water. And we could tell that because of there's marl in these samples, which indicates the slightly deeper water. But again, we see that among the difference here is driven by silt and deep water. Okay, so far what I've shown you here, there's definitely a drop in diversity that might correspond to the mass extinction event, and that there's these changes in lithology and changes in environment that are clearly controlling the differences in the taxonomic composition among these. And so there's one more check we can do to assess whether or not this is truly an extinction event here. And so what I did was I took all the brachiopod genera that I sampled, and I went to the paleobiology database to determine when exactly did they go extinct at the global scale. And so I plotted them here. And so this is a very rough figure, but basically on the y-axis here, you get the geologic stages and geologic age. And in this case, and up here on the x-axis, you get the genera of brachiopods that I sampled. And so each of these bars correspond to how long they were around in the known fossil record. And the red bar here corresponds to the Mississippi and Pennsylvanian boundary. And so what I want you to know is that for basically every single genera of brachiopods that I sampled, only one actually goes extinct at the Mississippi and Pennsylvanian boundary. Everything else either crosses the boundary or originated in the Pennsylvanian. And so, and just to note, this figure is a little bit outdated, but you can see these three genera here that just seem to just barely cross. They actually end up being closer to up here after there's new data that was uploaded to the database. But this genera here, Protactella, it's the only one that truly went extinct globally. And so to sum up, based on what I saw at Arrow Canyon, there's no evidence of an extinction event, at least observed within the brachiopods. And so all perceived changes in these fossil communities are driven by lithology. And so one of the ways too that we can account for this loss of this apparent loss of extinction is to apply a sequence stratigraphic approach. And so at the time that I was working on this, I was not really familiar with sequence stratigraphy, and so I didn't. But if I were to go back and be able to sample multiple locations in multiple sections, it's possible that we might see where that apparent loss of brachiopods from the lower part of the section that perhaps are found somewhere else because they migrated to wherever their environment, where the preferred environment was located. And so, and here just one example of how understanding sedimentology and stratigraphy is important to understand biological patterns, because at first glance, you might see there's definitely evidence of an extinction when in fact there actually is not. All right, so now I'm gonna shift gears here, and I'm gonna talk about my work that I did during my dissertation. And so for my dissertation, I basically did a lot of work on the Jurassic and the Meso-Jurassic. And in particular, part of my dissertation focused on the Sundance Seaway, which you see here. And the Sundance Seaway was this very elongated epicontinental seaway that inundated the western margin of North America from the middle, particularly in the middle Jurassic to the very early late Jurassic. And the Sundance Seaway is the precursor of the better known Western Interior Seaway during the Cretaceous. And so the Sundance Seaway is very interesting because it's is quite fossiliferous, but not many workers have worked on it, with the exception of a few workers who they've done a lot of work on plesiosaurs that are preserved in the Sundance Seaway, which are found in the Bighorn Basin here in northern Wyoming. And so the Sundance Seaway was also interesting because as you can see here, sort of this very long seaway that only has one entrance. And so one thing we noted from our lab group in Georgia, at the University of Georgia, was that you can see evidence of an environmental gradient as you go along, as you move latitudinally on the seaway. So basically, as you go further north, you see evidence of more marine conditions. And as you go further south, it becomes more restricted. And I'll just put up a couple of arrows here so you can see. As you go further south, you get lots of deposits of gypsum and other evaporites. And so clearly, it was more saline. It was, in some cases, it was hypersaline in some areas. And this is also noted by the organisms that live there. And so one of the things that we wanted to see is, okay, well, can we see true evidence that the biota of the Sundance Seaway is tracking this environmental gradient? And so you would expect that as you go further south, you would lose diversity. It will affect the ecological composition of the communities. And so what I did was I focused my work on the southern part of the seaway in Utah. You can see down here. And so a postdoc and a grad student working in our lab focused primarily in the Wyoming area of the seaway. And so they collected, they both used a sequence stratigraphic approach to collect paleobiological samples and to assess the paleobiology within that sequence stratigraphic context. So I did the same thing in Utah. And so now what I wanted to do was compare using that approach and see how the biology changes, see how the biota changes. And so in the Wyoming part of the Sundance Seaway, primarily the units of focus were the Gypsum Spring Formation and the Sundance Formation. And so in the South, however, the equivalent units to the Sundance are not there. And so the reason why is the Gypsum Spring Formation was deposited during the Bajocian and Pathonian stages of the Jurassic, but the Sundance is a little bit younger. And during the formation, during that time, the seaway did not expand as far south. And so our units in the South do not have the marine units you see up in the North. And so what I did was I compared this part of the Sundance, of the seaway up north to what I saw in Utah. And so the Bajocian-Bathonian interval of the Jurassic is important because globally, we see a huge change in diversity. We actually see a rapid rise in diversity. And not just that, but we also see major changes in ecology. And so during this time, it's the beginning of what's known as the Mesozoic Marine Revolution, where you start seeing ecological adaptations that are potentially being driven by escalation interactions in which predators and competitive species are driving adaptive evolution. And so you see that globally. And so this is a great interval to see if we can see those same patterns, but at a more regional scale, and also by controlling for the environment. And so by using a sequence stratigraphic approach, we can control for water depth, because water depth is a primary driver that controls variation in composition, because organisms prefer different water depth. And so you're going to find them living in different places along a water depth gradient in the ocean. And so I used the framework that Annika Clement and Sylvia Denise used, and went down to the South to measure the sections, create the same framework, and collect samples over there. And so what they did in the North, they sampled along a gradient. And so they have about 12 sections they measured, and they created a correlation. Here is just one section as an example. In the South, in Utah, I measured 7 sections, but instead of creating the correlation, I just created one composite section, and so which is you see here. And so what the cool thing is, we can actually correlate the two. And so the equivalent here, or the datum we can use to compare, is actually this interval right here, where you see the Sapka facies go into limestones. You can see them here as well. And so I use some of the samples I collected from down here, which are equivalent to the ones I collected here, and the ones up here, which they're equivalent to ones here. And so what I wanted to do is, again, only look at same facies, same water depth. So in this case, it's open shell subtidal, to assess how ecology changes and how diversity changes along the seaway, and if it relates to global patterns. So first off, I'm going to start off with diversity or richness. And so here what I'm showing you is a couple plots using a method called additive diversity partitioning, which is a way to break up diversity. Here you see mean alpha, beta, and gamma. Alpha just means the average number of genera or species in a sample. Beta means the diversity between samples, or the difference between samples. And then gamma is the overall diversity. So in the south, diversity doesn't really change as you go up section, so as you go through time. So as you go from the Bajocian to Bathonian, it's pretty much the same. But when you compare it to the north, you see in the north, there's a huge jump in diversity. And so what I should note here is, again, so I mentioned that I compared the diversity and ecology among the same facies, but also the sample sizes are the same. So very similar samples, number of samples, and very similar number of individuals assessed. And so even when you take that into account, diversity was still a lot higher in the north. It does drop a little bit during the Bathonian, but I'll get to that in a little bit as to why that might be it. But the take home here is diversity is definitely a lot higher in the north. So now I'm going to compare the ecology. And so basically what I did was I took all the fossils, all the marine organisms that were living there, and put them into their ecological categories and compare them between north and south, and also from Bajocian to Bathonian. And so here, the way to read these plots is this represents the difference in proportional abundance. So basically, among these ecological groups, you can see here on the x-axis, if you see a dot here, this point is above the 0 line, that means that these groups are more abundant in the north relative to the south. And if you see these points below the 0 line, they're negative, that means that in the south, they were more abundant. And so you can see here, they're more or less the same. It's you don't see a big difference, at least during the Bajocian. But when you look at the Bathonian, so the stage, the younger stage above it, there's a huge change, and particularly in cementing and epiphaneal organisms. So cementing used to be more dominant in the south during the Bajocian, but now they're way more dominant in the north. And so, but everything else more or less stays the same. And what's interesting is that you would expect that that they will remain the same because the gradient doesn't change. But it turns out that the difference, the big thing that's driving these differences is actually lithology again. And so again, I'm looking at the same water depth, but when you take a closer look at the lithology, they're a little bit different. And so during the Bajocian in the south, you see here how they're very shelly, and these are just chock full of shells, and there's hardly any silt, any mud in between the shells. I know here it seems that way, but this is just the covering on the surface of the bed. If you look within the sample itself, there's hardly any mud or silt. But in the Bathonian, the opposite is true. You get a lot more mud in the matrix. And so here you can see a few of these same shells, the same oyster, in this case is Lyostria. And you can see how there's much more mud involved. And so here, basically in the south, cementers were more dominant during the Bajocian, less so in the Bathonian. And so the same thing happened in the north, but in reverse. And so in the north, during the Bajocian, the same water that facies was more muddy, but in the Bathonian, it became more shelly. And so to sum up, there's no major changes in ecology spatially, but with the exception of cementers and epiphaneal groups. And these differences are entirely driven by changes in shelliness. So water depth doesn't play a role here. It's just entirely the lithology. So, and basically, there's just more mud during there's more mud during one interval relative to the other, and they basically flip flop between the north and south. And so, which is pretty cool, and something we didn't expect. And it doesn't show that same gradient you would expect, again, because even though you have those environmental conditions, you still have the sedimentology that's playing a role and ultimately controlling at least the preservation of these ecological of these biotic communities. Okay, so far I've shown you some examples of how sedimentology and stratigraphy affect how we view biology using field data and field approaches. And now I'm going to focus on the body size stuff that I mentioned at the beginning. And so here at Stanford, my advisor, John Payne, has been putting together a giant data set of body size data for marine animals. And this includes everything from very tiny protists and foraminifera and even viruses all the way up to vertebrates, whales, and marine reptiles as well. And just to understand what's driving body size patterns throughout the Phanerozoic. So specifically what I'm doing for this work for my postdoc is I'm assessing how body size selectivity might change between background intervals and during mass extinction events. So selectivity is just basically determining what traits are driving increased extinction in certain groups. And so in this case, I'm using body size. And this stems from this idea of that goes back to the 80s, in which mass extinction intensity. So we know that mass extinction intensity is greater during mass extinction events than during background intervals. And you can see here in this plot where these spikes here correspond to the big 5 mass extinction events. And when you compare it to background intervals, you see here shaded in green to see how the intensity is much higher during mass extinction events. And so the question that I'm asking then is, does selectivity do the same thing? So if the selectivity during background intervals, so say for instance that during background times, larger body size corresponds to higher extinction, does that same pattern hold during mass extinction events, or does it change? And that's what I'm focusing on. And so body size and extinction has been talked about for a long time in paleontology. As a matter of fact, this idea first goes back to the 1800s. And it was made by observation that at the end of the Pleistocene, you have the extinction of the megafauna. And so you have very large organisms that went extinct. But also when you look at other extinction events like the one thing at the Cretaceous, you see that dinosaurs went extinct. And so there's this long-held idea, at least looking at terrestrial vertebrates, that large body size corresponds to higher extinction risk during mass extinction events. And so this idea has been around for a long time. And so in order to test that, my postdoc advisor, John, and a former postdoc of his, Noel Heim, tested this idea, but using the marine record. And what they did was they took body size for all the major marine invertebrate groups that we have throughout the Phanerozoic, and they assessed the extinction risk based on body size, starting at the early Ordovician all the way to the Pleistocene. And so here are the results of their study. And so basically the way to read this plot is these are coefficients from a logistic regression analysis conducted using capture mark recapture modeling. And so the way to read this is coefficients that are positive correspond to large body size associated with increased extinction risk, whereas those that are negative, of course, means that small body size is associated with extinction risk. And so as you can see here, for the majority of groups, for all groups, or the majority of all groups they looked at, they found that smaller body size on average means you have an increased extinction risk throughout the fossil record. Now, this doesn't take into account mass extinction events. This is just what we would consider background time. And so throughout the entire, well, the majority of the Phanerozoic, you see this pattern. And so one of the things that we're also considering is the sampling. And so I mentioned about the stratigraphic record and how that's important in understanding biological patterns. And so when you assess diversity or any macroevolutionary patterns across the Phanerozoic, the big issue we're always going to come across is sampling because of the incomplete stratigraphic record. And this is, and this idea of the incompleteness of the stratigraphic record with regards to evolution goes back to Darwin, in which Darwin offered dismay that we may never truly get a sense for the evolutionary dynamics because there's so many gaps in the fossil record. But this method of capture marker capture, which I'll go over in a little bit, accounts for that. And so it accounts for that sampling. And so in here, what this suggests that when you take sampling into account, and so I mentioned about the stratigraphic graphs, but also the other issue is human bias when it comes to collecting samples. And it's a well-known bias that most paleontologists tend to collect the larger specimens when they go on the field. And so, and what this figure on the right shows, that for the majority of these groups, there's a bias against large body size. And so here basically is that, so smaller body size means, the coefficients, negative coefficients mean that small body size is samples less. And so even when you take that into account, you see this pattern of small body size associated with increased extinction risk. And so just to go over a little bit about where the data comes from that they use, and that I also use for my study. And so let me go back a little bit. Yeah, so I mentioned here small body size linked to increased extinction risk, and how that contrasts. So now hopefully you see that it contrasts to large body size associated with increased extinction risk for vertebrates during mass extinction events. And so what I wanted to assess was, okay, does this change during mass extinction events? And so now I'm going to use the same approach, but then add mass extinctions to it. And so the body size data that we have comes from the summer program that Rowan mentioned at the beginning that I helped run, which high school students come in and collect data, body size data. And in the process, they get to learn about different natural history aspects. We've worked on the Ordovician, on the Ediacaran, worked on the Pleistocene. And so they get to work on so many different cool projects. And all the while they collect data and they use that same data. And so this summer was the first time I ran the program. And because of the COVID pandemic, we couldn't collect data, but we were able to do some cool projects nonetheless with the students. And so, but now to move on to the actual data, the data set I use is a big data set that consists of a little over 10,000 genera of the big major invertebrate groups, arthropods, brachiopods, echinoderms, and mollusks, but I also have data here for bony fish. And again, using the same approach that John and Noel used, I focused on the early Ordovician to the Pleistocene to assess how selectivity with body size changes. But I didn't just use extinction as well. I also focused on origination, because origination is also an important component of extinction. And so how things originate and bounce back after mass extinction events is important to understanding evolutionary patterns and outcomes. And so just real quickly to go over capture mark recapture. And so the way this method works is very similar to how you use in ecology. You go out during your first sampling attempt, and you take a census of every organism, either individuals or species you find, and make a note of it. And then you come back several times after to see if you can still track those original species or individuals. And so every time you come back, there's a couple probabilities you need to worry about when trying to assess the number of individuals or taxa. So N here represents the number of taxa that were first encountered during the first interval. But in between intervals, we have to consider the probability that they survive, because they may not survive between encounter attempts, or the probability that even if they did survive, that you would encounter it. And so oftentimes, if you're tracking organisms, even if you survive, you're not going to see them again when you try and look for them. And so you use this approach on the fossil record to assess extinction and origination on body size. And for us, these time intervals here corresponds to every geologic stage from the Ordovician to the Pleistocene. And just to show you the results here real quick, first I'll go over extinction. And what this it's very pixelated, okay? So what this shows here is you see these squares and triangles. These represent background extinction levels. And so very similar to the study by Payne and Heim, smaller body size is associated with extinction risk during background times. But during mass extinction events, that changes a lot. And for most of these groups, we see that larger body size is now linked to increased extinction risk for most groups, except for one, which are other crinoids. So it follows that same pattern we see with the vertebrates, that mass extinction events definitely do drive extinction of larger body size. And so when you take a look at origination, we see a pretty cool, interesting pattern as well. And so during normal background times, most groups originate small, which makes sense, with the exception of ostracods. But after a mass extinction event, how they come back it differs quite a bit. And so there's this group here, primarily of ranulated brachiopods, crinoids, and cephalopods tend to originate smaller after mass extinction events. But mollusks here and gastropods and bivalves, bony fish, and ostracods tend to come back bigger. So what's driving these differences in responses to body size to extinction and origination? I'm not 100% sure, but it's one thing that I'm working on right now, and to try to assess those patterns. So in summary, there's strong evidence that of alternating macroevolutionary regimes. So in other words, selectivity changes between background intervals and mass extinction and the recoveries for most groups of marine animals we looked at. Origination has a much stronger effect than extinction itself. If I go back here real quick, you see that for the most part, these origination coefficients tend to be much greater than the extinction ones itself. And so origination is definitely playing a more stronger role than extinction is. And not all classes alternate in the same direction, which means there's other underlying factors that might be driving that. And the last thing is to go back to the main theme, capture mark recapture accounts for sampling issues to assess for body size evolutionary patterns. So we get control for those stratigraphic gaps for how stratigraphy may potentially impact affect how we view evolutionary patterns in the fossil record. And so with that,